Steps in initiation. ORC binds in the ORIC site of the DNA. Recruits CDE6. CDC6 binds with ORC in ATP dependent manner. CDT1 binds with C terminus of CDC6. CDC1 is required for licensing the chromatin for replication. CDC6 recruits the CDT1. So CDT1 binds with C terminus of CDC6, finally binding all three protein recruit MCM. MCM finally binds with chromatin. These following steps occur in G1 phase of cell cycle. This is structure. G1 phase, MCM, CDT1, CDC6 and ORC MCM CDT1. Pre-S phase, CDT1, CDKS and DDKS will be there. So CDC beta ORCS. S phase, CDC4, GINS, CDC4 and ORCS is there. The activity of CDT1 during the cell cycle is regulated by a protein called geminin. It also inhibits CDT1 activity during this phase in order to prevent the re-replication of DNA, ubiquitination and proteolysis. Initiation complex. It is the second stage in DNA replication where the pre-replicative complex is converted to initiation complex. Involved protein cell division cycle 45, CDC45, GINS, then cyclin dependent kinase or CDK, DBF4 dependent kinase, DDK, DBF4 dependent kinase or DDK, combination of CDC7 and DBF4. Steps in initiation complex. CDC45 protein is a compound which is needed for conversion of pre-replication complex into initiation complex, CDC45. It binds with chromatin after the beginning of initiation in late G1 phase by physically associated with MCM5. It binds with chromatin after the beginning of initiation in late G1 phase by physically associated with MCM5. The binding of CDC45 is based on CLB, CDC28 as well as the function of CDC6 and MCM. Genes are essential for interaction of MCM and CDC45 at ORI site, origin of replication site C, during initiation. Once the initiation complex is formed and the cells pass into S phase, the complex then becomes a replisome and elongation is initiated. Once the elongation is initiated, it forms the replication fork by unwinding the DNA strand. As the double helix of DNA separates from one side and supercoils are formed on the other side. The problem of supercoils comes in the way of DNA replication is solved with the help of a group of enzymes called DNA topoisomerase. Replication fork. The replication fork is the junction between the newly separated template strand known as leading and lagging strand and double-stranded DNA. Elongation occurs in the 5' to 3' direction in both the leading and lagging strand. Five prime to 3' direction. So we could see 5', 3' strands. At the replication fork, many DNA replication enzymes assemble to form the replisome. Topoisomerase 1, topoisomerase 2. Leading strand is there, lagging strand is there. MCM helicase will be there. Then primase, whole alpha, RPase, primer formation, ligase, Okazaki fragments. Leading strand is also there. Leading strand. The leading strand is the template strand that is being replicated at the same direction as the movement of the replication fork. Nucleotides are added by the DNA polymerase. DNA polymerase requires the delta. DNA polymerase requires the DNA primer produced by primase. DNA polymerase requires the RNA primer produced by primase. Elongation takes place in 5' to 3' direction. Finally, the primer are removed by RNase H and gap is sealed by the RNA ligase 1. Finally, the primer is removed by RNase H and gap is sealed by DNA ligase 1. Finally, the primer is removed by RNase H and the gap is sealed by DNA ligase 1. Lagging strand. DNA replication on lagging strand is discontinuous and elongation in opposite direction to replication fork. Nucleotide is added by the DNA polymerase delta. Lagging strand used more RNA primer for loading nucleotides. DNA polymerase will synthesize short fragments of DNA called Okazaki fragments, which are added to the 3' end of the primer. These fragments can be anywhere between 100 to 400 nucleotides long in eukaryotes. Then termination. The termination of replication in eukaryotic cells occurs by telomere regions and telomerase. Telomeres extend the 3' end of the parental chromosome beyond the 5' end of the daughter strand. This single-stranded DNA structure can act as an origin of replication that recruits telomeres. Telomerase is a specialized RNA polymerase that consists of multiple protein subunits and an RNA component. The single-stranded DNA structure can act as an origin of replication that recruits telomerase. Telomerase is specialized DNA polymerase that consists of multiple protein subunits and an RNA component. The RNA component of telomerase anneals to the single-stranded 3' end of the template DNA and contains 1.5 copies of the telomeric sequence. Telomerase contains a protein subunit that is reverse transcriptase called telomerase reverse transcriptase or TERT. Telomerase reverse transcriptase. Five prime to 3' direction. Telomerase. The binding polymerization translocation cycle can occur many times. This greatly lengthens one of the strands. Step 1 binding. Step 2 polymerization. Step 3 translocation. Eukaryotic DNA telomerase synthesizes 6 nucleotide repeat. Telomerase moves nucleotides to the right and begins to make another repeat. The complementary strand is made by primase, DNA polymerase and ligase. Inhibitors of DNA replication. Fluorodeoxyuridylate. Fluorodeoxyuridylate. 5-fluoro-2-deoxyuridylate an inhibitor of thymidylate synthetase formed from 5-fluorouracil or 5-fluorodeoxyuridine. Overview of 5-FU. Fluoro plus uracil, uracil, thymine, DNA synthesis, cell proliferation. Fluoro plus uracil, uracil, thymine, DNA synthesis, cell proliferation. 5-fluorouracil. Fluorouracil is a pyrimidine analog and anti-neoplastic anti-metabolite, interferes with DNA synthesis. Fluorouracil. The structure of uracil and 5-FU, 5-fluorouracil and uracil. We could see the structure. Then the role of uracil. A precursor of thymine via thymidylate synthetase. Part of composition of RNA. The role of uracil. The precursor of thymine via thymidylate synthetase. Part of composition of RNA, synthesis of proteins, in particular cellular enzymes, uracil. So 5-FU blocks the methylation of uracil. 5-FU blocks the methylation of uracil towards thymine. 5-FU blocks the methylation of uracil towards thymine. Thymidylate synthetase, we could see uracil, thymidylate synthetase, thymine. 5-FU blocks RNA transcription. Uracil, 5-FU RNA transcription is blocked. So in summary, 5-FU inhibits DNA synthesis and DNA transcription. 5-FU inhibits DNA synthesis and RNA transcription. Next, methotrexate. Methotrexate overview. Folate, methotrexate, DNA or RNA production decrease, cell proliferation decrease. Methotrexate is a type of anti-metabolite. Both healthy and cancerous cells use folate to make DNA and RNA. Both healthy and cancerous cells use folate to make DNA and RNA. Methotrexate interferes with this process, helping to stop the production of cells. So cancer, lung cancer, breast cancer, anti-metabolite methotrexate will be interfering with that. Then rheumatoid arthritis also, DMARD, then anti-inflammatory, psoriasis, Corin's disease. Methotrexate acts as a drug. Folic acid and methotrexates are very similar in structure. Folic acid and methotrexate. Folic acid, Pg, FH2Pg, FH4Pg. DHFR and DHFR will be the one carbon. Methotrexate, MTXPG, TTMP and DUMP. Structure of methotrexate. Pteridine ring, PABA and glutamic acid. Methotrexate competitively inhibits the enzyme that works for folate metabolism. Methotrexate competitively inhibits the enzyme that works for folate metabolism. Folate enzyme methotrexate, DNA synthesis decreases, cell reproduction decreases. Role of folate coenzyme in DNA synthesis. Folic acid, dihydrofolate, tetrahydrofolate, 5,10-methylene THF, 5-methylene THF, then 5-formyl THF, purines, 5-methyl THF, pyrimidine. DNA synthesis, DNA methylation, methyl group donor, 1 carbon unit, methionine is there. Then role of methotrexate for cancer therapy. Folic acid, dihydrofolate or DHF. Folic acid and dihydrofolate or DHF. DHFR, dihydrofolate reductase, tetrahydrofolate. Methotrexate completely inhibits DHFR. Dihydrofolate reductase, methotrexate. The affinity for methotrexate for DHFR is about 1000-fold that of folate. Role of methotrexate for cancer therapy. DHFR binding will be happening, so tetrahydrofluorate is not formed. DNA methylation decreases. Next topic, rolling circle model of DNA replication. Introduction. A rolling circle replication is a process of unidirectional nucleic acid replication. Can rapidly synthesize multiple copies of circular molecules of DNA or RNA, such as plasmids. Eukaryotic DNA also replicate, widely used in molecular biology and biomedical nanotechnology, especially in the field of biosensing as a method of signal amplification. So steps, circular dsDNA will be nicked. Circular dsDNA will be nicked. 3' end is elongated, that is leading strand. 5' end displaced, lagging strand. Made up of double-stranded by Okazaki fragments. Replication of both uninicked and displaced SS DNA. Displaced DNA circulates and synthesizes its own complementary strand. So first circular DNA is there, nick is formed. New DNA synthesis, displaced strand. Synthesis of complementary DNA strand like this. Replication in eukaryotes is bidirectional. This type is unidirectional. Ideal example of this type is the circular plasmid in bacteria, as it happens only in circular genomes. Initiation initiates phosphatase by the action of helicase topoisomerase, single-stranded binding proteins SSBPs. Initiation initiates phosphatase by the action of helicase topoisomerase single-stranded binding proteins. Next, elongation. For elongation, OH group of broken strand using the unbroken strand as a template, the polymerase will start to move in a circle for elongation, due to which it is named rolling circle model. End will be displaced and will grow out like a waving thread. You get the structure. DNA polymerase 3 is there. Broken thread, origin point. This is rolling circle DNA replication. Then termination. At the point of termination, the linear DNA molecule is cleaved from the circle, resulting in a double-stranded circular DNA molecule and single-stranded linear DNA molecule. The linear single-stranded molecule is circularized by the action of ligase and the replication of double-stranded circular plasmid molecule. Conjugation of F+ and F- bacteria, another topic not related to this one. Donor bacterium F+ cell, then F- cell conjugation process. During conjugation, one cytoplasmic connection forms between the F+ and F- cell. One of the DNA strand on the F acts as nicked and separate the F factor is inserted during conjugation between F minus and F plus cells. This is an example. So rolling circle replication, ORI rep A 3 prime OH region 5 prime. Replication starts at ORI region by a initiator replication A. DNA polymerase 3 region binds with the DNA polymerase and replication starts. Helicase is also attached to the region. Single strand binding protein. Progeny positive strand synthesis. DNA helicase joins that. Positive strands also synthesis. It's complementary synthesis. Breakage is joined by helicase. Progeny strand can also synthesize another strand of DNA. Overall explanation of rolling circle. Nick formed new DNA synthesis. Displaced end. Synthesis of complementary DNA strand. Next topic, process of DNA replication, theta model. Initiation of replication occurs at a specific region called origin of replication, where the dsDNA denatures to form ssDNA and within which replication commences. The locally denatured segment of DNA is called the replication bubble, and the two strands in this region using which new complementary strands are synthesized are called the template strands. As the DNA unwinds, a Y-shaped structure is formed at either ends of the replication bubble. This is known as replication fork. As the DNA unwinds, Y-shaped structure is formed at either side of the replication bubble. This is known as replication fork. In such cases, bidirectional replication occurs. The fork is generated by a complex of 7 proteins called proteasome that includes DNA G primase, DNA B helicase, DNA C helicase assistant, DNA T and primase A, B and C. In coli, Escherichia coli, the ORIC region spans 245 base pair and contains clusters of 3 copies of 13 mer and 4 copies of 9 mer sequence. To initiate replication, an initiator protein called DNA AATP, encoded by DNA A gene, binds to 9 mer sequences and denatures the region connecting to it to 13 mer sequence by breaking 8 T bonds, which are weakened as they are held by 2 hydrogen bonds. This requires energy from ATP. This forms the initial complex. DNA helicase, DNA B is loaded onto the DNA strands by helicase loader DNA C. DNA helicase untwists the DNA. This forms the pre-priming complex. SSBP, single-stranded binding protein, binds the open strands of DNA to prevent rewinding. Gyrase, a type of topoisomerase, releases the tension generated by rapid unwinding of the DNA strands at 3000 RPM. The DNA helicase recruits primase enzymes that synthesizes a short segment of 5 to 10 nucleotides called primer, which allows elongation of DNA. This happens because DNA polymerase 3 can only add nucleotides but cannot initiate synthesis of a new strand. Elongation is carried out by replisome, which is made up of DNA polymerase 3 and the primosome complex. The DNA pol 3 enzyme tethers itself to the ssDNA via its core enzyme. The core enzyme catalyzes DNA synthesis by adding complementary nucleotides. DNA synthesis is taken place in 5' to 3' direction towards the replication fork. The strand that is being synthesized in this direction continuously is called leading strand. while the strand that is synthesized in the opposite direction is called lagging strand. The leading strand requires just one primer, whereas the lagging strand requires many such primers. Since leading strand is synthesized continuously and simultaneously along with the discontinuous synthesis of lagging strand, the entire process of DNA synthesis is a semi-discontinuous process. Fragments of lagging strand are known as Okazaki fragments. After the strands have been completely synthesized, these Okazaki fragments are joined together. DNA polymerase 3 is removed. DNA polymerase 1 removes the primers by its 5' to 3' exonuclease activity, exposing the template nucleotides. It then adds complementary nucleotides by its 5' to 3' polymerase activity to the 3' OH end of the previous Okazaki fragments, thereby replacing the primers. The next that remain behind are joined by ligases by creating a phosphodiester bond. This is known as Nick translation. Any errors in base pairing is removed by DNA polymerase 3 by its 3' to 5' exonuclease activity immediately before proceeding on to the next nucleotide. This is called proofreading. Then termination of this process occurs when the replication fork reaches the termination sites. TUS proteins, terminus utilization substances. Terminus utilization substances bind to TER sites. TUS proteins or terminus utilization substance bind to TER sites and halt progression of forks. Equally, there are 10 replication termini, each spanning 23 base pairs. TER B and C terminate the clockwise fork, while TER A, D, and E terminate anti-clockwise fork. In circular chromosomes, the daughter chromosomes remain interlocked and are called catenans. Topoisomerase 2 involves this problem by breaking some bonds in DNA molecules so as to separate the strands. Decatenation. Replication fork, newly formed strands, old strands. Theta model of prokaryotic replication. Restriction endonucleases, next topic. Restriction endonucleases are enzymes that produce internal cuts called cleavage in the DNA molecule. Enzyme that cuts DNA at or near specific recognition nucleotide sequence is known as restriction sites. That is, the enzyme recognizes a particular target sequence at a duplex DNA molecule and breaks the polynucleotide chain within that sequence to give rise to discrete fragments of defined length and sequence. Restriction enzymes are commonly classified into 3 types, which differ in their structure and whether they cut their DNA substrate at their recognition site or if the recognition and cleavage site are separate from one another. Restriction enzymes are commonly classified into 3 types, which differ in their structure and whether they cut their DNA substrate at their recognition site or if the recognition and cleavage sites are separate from one another. Recognition site, cutting site, treatment with EcoRI, complementary tails are formed. To cut the DNA, all restriction enzymes make 2 incisions, once to sugar phosphate backbone and of each strand of DNA double helix. These enzymes are found in bacteria and archaea and provide a defensive mechanism against evading viruses. Recognition site. Restriction enzymes recognize a specific sequence of nucleotides and produce a double-stranded cut in the DNA. The recognition sequence can also be classified by the number of bases in its recognition site, usually between 4 and 8 bases. The amount of bases in the sequence will determine how often the site will appear by chance in any given genome. 4 base pair sequence would theoretically occur once every 4 raised to 4 or 256 base pair. 6 bases 4 raised to 6 or 4096 base, and 8 bases would be 4 raised to 8 or 65535 base pair. Many of them are palindromic, meaning the base sequence reading the same backward and forward. In theory, there are 2 types of palindromic sequences that can be possible in DNA. The mirror-like palindrome is similar to those found in ordinary text, in which a sequence reads the same forward and backward on a single strand of DNA, such as GTA-ATG. The inverted repeat palindrome is also a sequence. Mirror palindrome, mirror-like palindrome. Next, the inverted repeat palindrome. Inverted repeat palindrome is a sequence that reads the same forward and backward, but the forward and backward sequences are found in complementary DNA strands of double-stranded DNA. That is, GTATAC being complementary to CATA-TG. The inverted repeat palindrome. First, the mirror-like palindrome, the inverted repeat palindrome. The mirror-like palindrome and the inverted repeat palindrome is also a sequence that reads the same forward and backward, but the forward and backward sequences are found in complementary DNA strands. Double-stranded DNA. Types of ends produced using restriction enzymes. EcoR1 digestion produces sticky ends. Types of ends produced using restriction ends. EcoR1 restriction digestion produces sticky ends. DNA fragments with complementary sticky ends can be combined to create new nucleotides, which allows the creation and manipulation of DNA sequences from different sources. Whereas SMA1 restriction enzyme cleaves produces blunt ends. These blunt-ended fragments can be joined to any other DNA fragment with blunt ends. Enzymes useful for certain types of DNA cloning experiments. Recognition sequences in DNA differ for each restriction enzyme. Recognition sequences in DNA differ for each restriction enzyme, producing differences in the length. Restriction sequences in DNA differ for each restriction enzyme,